So chromatography is a method of using what's called chromatography paper to separate a mixture of solutions based off their polarity. So this is just a simple setup here. Let me use a different color. Simple setup of chromatography paper taped to a popsicle stick, because all that matters is that the chromatography paper is vertical. And what we're going to do to this chromatography paper is we're going to submerge it into a solution. In this case, we're going to use a polar solution. You could use water, but we're going to use isopropyl alcohol. Isopropyl alcohol. And that isopropyl alcohol is going to, as it touches the bottom of this paper, seep up through the paper. Now, if there were anything on this paper that were polar, for example, since isopropyl alcohol is polar, it will carry the polar substances up with it. The more polar a substance, the more it will get carried upward towards the top of the chromatography paper. So in this quick lab, we use the 7 food dyes that the FDA has approved for the use in foods, and we have set up a chromatography setup for each one of them. So over here we've got yellow 5. You can see that it's been submerged in the chromatography paper, and the yellow food dye number 5 has moved up the chromatography paper, leaving a slight trail at the bottom, but the majority of it is up at the top. Now these are the in-progress pictures, so they're not quite finished. For red 3, we can see that the red dye has moved up the paper, but you can see right here that the solvent has gone all the way to that red line, but it has not carried the dye with it up that far. So that gives us some indication that red 3 is not as polar as some of these other dyes are. Here's red 40. Right here on this line is where the solution has made it so far. Again, these are in-progress pictures, and you can see the red dye is in line with that. The isopropyl alcohol is doing a very good job of carrying that red dye, so that red dye 40 must be quite polar. As for blue 2, it's kind of hard to see in the picture, but right here is where the solvent has reached so far, and you can see the blue dye has traveled upward with it, but has not quite gone as far as the solvent, so it might not be as polar. Again, yellow 6, right here along this line is where the solvent has made it so far, and the yellow dye is right there with it, so yellow 6 appears to be pretty polar. And the same can be said for green 3. The solution has made it to this point right here, and the dye is traveling with it. And our last one is blue 1, which the line is right here, and it is traveling with it as well, so blue 1 appears to be pretty polar. So what I did in this lab is I took 2 unknowns, which were simply a mixture of any of the 3 other dyes, and we performed a chromatography experiment on them to see how they would look. Again, these are in-progress pictures, but you can see in the back, this unknown A and unknown B both appear almost black because it's just a mixture of 3 of the other dyes. But you can see as you begin running a chromatography experiment on them, they'll start sorting out the dyes that make them up. So again, these are in-progress pictures, but we can see, especially in this unknown B, where there must be some sort of red in there and what looks like some sort of blue or maybe green. So these are actually after pictures, after the entire experiment has had time to run, and I chose to use experiment B simply because it worked better. So up here we've got green 3. Its chromatography, or its ink rather, traveled very far up, all the way with that solvent. That says to us that green 3 is really quite polar. This red 3, it did travel upwards with it, but notice how a significant amount of it stayed down at the bottom. And then this yellow 6, again, almost all of it traveled up to the top. So if you look at the unknown, you can see that it's got a long pink band, which matches the band of red 3, a high yellow band, which matches the band of yellow 6, and a high blue band, which matches the band of green 3. So by analyzing the chromatogram, which is just the name for these papers after you've run the experiment, after analyzing the chromatogram and matching up, that unknown B consists of the chromatograms of yellow 6, red 3, and green 3, we can conclude that unknown B was a mixture of green 3, red 3, and yellow 6. And here's just one more I wanted to show you. This is the finished chromatogram for red 40 and red 3. Ignore the fact in red 3 there's some blue dye. I accidentally set that down on the table and some blue, but oh well. You can see that the red of red 40 left a faint trail, but more of it traveled up, meaning there's not as deep of a color down at the bottom. Whereas in red 3, more of it stayed down at the bottom. Since our isopropyl alcohol was polar, it would make sense that red 40 would be more polar because it carried more of the red dye up with it. And if we look at the structural diagrams of red dye number 3 and number 40, we can see that red number 40 has an OH group, which means it's capable of hydrogen bonding, whereas red dye number 3 is not. So even though red dye number 3 is polar, red dye number 4 is capable of hydrogen bonding to that isopropyl alcohol. So it makes sense that red dye 40 would travel up better than red dye 3.